Thank you for inviting me. Look, I'm a kind of simple sort. I come from farming stock, and I try to work with things that work, basically. And I try to work within the limitations I've got. With the greatest respect to Robert and Claudius, I could spend my whole life complaining that the world's not a perfect place, and the drug companies are taking us over, and so on and so forth, and I'd end up disappearing up my own whatever. And so, therefore, I try to work with the system as we've got it, and have made two major contributions internationally, which I'm going to talk to you about. Now, first, a little bit of background. I'm a surgeon by training. My research degree was in embryology. I spent a lot of time in China in the late 1970s, early 1980s. I essentially got traditional Chinese medicine, or acupuncture in particular, going in the UK, founded the British Medical Acupuncture Society. Now, the recommendations, now this is the reality, this is the real world, okay, which I'm operating in, and the rest of you are doing the same, hopefully. The recommendations I brought over for what acupuncture might be useful for in the early 1980s from my time spent in China are now in the National Clinical the National Institute for Clinical Excellence in their recommendations for acupuncture. That's taken 30 years, but it's actually happened. So the real world works like that. It's slow. We are actually making progress. So that's some positivity. Now, what about the Cancer Act? This is most interesting. All the private hospitals in UK and all the private oncologists in UK are all breaking the Cancer Act on a regular basis. All the big health insurance schemes are doing it in the press, on the radio and the television on a daily basis. When challenged, and Stephen's got some stuff I've got on this, they say, well, essentially what they say is the Cancer Act is not for the likes of us, it's for the likes of the lowly medical detritus, of which I'm part, and some of you as well, presumably. And so, therefore, it applies to us. That's essentially what they've said. I've got a lot more material on that. It's all highly explosive. It's confidential at the moment. If there was some serious investigation into that, then I might be interested in sharing that. Right. Now, what am I faced with clinically? I'm faced with mostly cancers, some sarcomas, which are usually end-stage disease. They've usually gone through chemotherapy, radiotherapy, surgery, and so on and so forth, and they're not better. They're reaching the end of their evidence base. And therefore, that's what I've got to deal with. Now, the first treatment approach, which I've very much developed, I've been doing this over the last 9 to 10 years. This has been, I've taught it to several large groups in China, major oncology hospitals there, who are now using this with great success. And also, there's a study being done by the chief urologist at the University of Melbourne, who came over to see me, and I taught him how to do it. He didn't do exactly the same protocol as me, but has nearly completed the study on Gleason 6, Gleason 7 prostate cancer with most encouraging results. Now, I'm going to talk about sonar and photodynamic therapy. Now, this is one of our publications. I'm the first person to publish on this internationally. We've done 2 papers, and this is our first case series of 115 patients, of which the mother of one of our participants here was involved, and she was a complete cure of metastatic non-small cell lung cancer, completely unheard of. She's alive and well, nearly 9 years down the road. There we go. If you don't believe me, ask. That's the reality. Now, this is the case series. This is the site where we did it. 115 consecutive cases. Worldwide, as of now, there have been 1,000 cases done. The treatment is still experimental. until it's gone through phase 2 studies and so on and so forth, which I've now got a credible way of raising the funds in order to do that. And that won't be expensive for this, perhaps in the order of $15–20 million, which is not a lot of money in terms of drug development. Now, what is it? How does it work? Is it safe? Is it effective? Ways of enhancing the effect. And it works through cell-mediated immune function, immunotherapy. Why is that important to most of you nutritionists here and doctors who work in this field and those of you who are doing acupuncture? The effects you are producing are almost certainly mediated through innate and acquired immunity. That's probably what's going on. There's great difficulties in patenting immunotherapy approaches for all sorts of reasons, which is why they haven't been developed as much as they might have been, but there's a lot of increasing interest. And then tumor oxygenation, which you're probably familiar with. Now, first of all, let's talk about photodynamic therapy. Sono-and photodynamic therapy is a development of this. Light energy producing chemical energy. And in the middle of that, there's a photosynthesizer. That's how photosynthesis works, basically. Without that, none of us would be alive. Now, it's the conversion of light to chemical energy. Chlorophyll is important. The compound we use is extracted from chlorophyll. It's extremely expensive to make because you've got a big lump of chlorophyll here, and then you end up with about that much of the solar and photosensitizer. That's why it's phenomenally expensive. This is that's the molecular shape. Actually, the molecular shape is quite interesting because it looks like a spring. That relates to its ultrasound sensitivity. Now, light-activated treatment has been around since ancient Egypt. Modern cancer treatment, light or light-emitting diodes, laser light or light-emitting diodes are used. It's used on surface tumors because light doesn't penetrate terribly far. It's becoming more and more integrated into standard health systems worldwide. It's a great treatment. Now, and that's the reference. Now, it's a multi-stage process. First of all, administration of the agent. Conventionally, these are porphyrins, which are related to hemoglobin. And the agent we use isn't a porphyrin, and our agent relates to chlorophyll. So it's a light-sensitive medicine. The standard treatments used now use porphyrin ring structures. We use a chlorophyll one. It's either given IV orally or topically, and it preferentially goes to tumor cells. It's activated by light, and it's specific to certain wavelengths of light. Red is the color most often chosen, and what happens is this. The mechanism of action is by tumor necrosis. That's cell death. The cells just disintegrate. Photosensitizer goes into the cell. Specific wavelength of light, usually about 660 nanometers, that's the wavelength. Activates the photosensitizer. That splits oxygen into singlet oxygen, which is highly reactive, and produces hydroxyl radicals and reactive oxygen species. And then as a result of that, the cell dies by necrosis. It actually disintegrates. And that produces an inflammatory response. And many of the cases we use have such a lot of tumor around, we have to cover them with a steroid, dexamethasone, often at quite high doses. One of our main problems with this treatment approach is that we get too much tumor cell death. Too much, okay? And we get tumor cell death in nearly 80% of the patients we treat. It's just truly astonishing, absolutely astonishing. This is why it's such an important treatment. Apart from that, there are no side effects. So the next thing that happens, there's an immune response. And that's the kind of response all of you are having with doing your various therapies and so on and so forth. Almost certainly, that could be studied, by the way, it's not difficult. And you get phagocytic cells, that's cells which gobble up bits of the dying cell. You get dendritic cells. Dendritic cells pick up the the molecule which is called the antigenic determinant, we call it the epitope, and it's only a small bit of the full tumour antigen, which says that's your tumour as opposed to somebody else's tumour. The tumour antigen is about 30,000 amino acids long. The epitope's only about 10 to 15 amino acids long, and it sits on a molecule called the major histocompatibility type 1 molecule, which differentiates you from your neighbour. And then that epitope, that code for your tumour, it's just like in a computer program. You put in a password and you get into the program. If you haven't got the password, you can't get in. And then the dendritic cells pick that up, and then they go and they activate in local lymph nodes tumour killing. That's what cytotoxic means. CD8 T cells. CD8 is the molecules on the surface of these white cells which differentiates them from other cells. And they become tumour specific, and they go in, they target your tumour, remaining tumour, and they use various enzymes and they destroy that tumour. It's absolutely specific. It's as precise as the kind of thing you get on a computer and all these security systems and whatever. It's as precise as that. So the host's immune response to the cancer can be assessed also by the number of these tumour-killing T cells within the tumour. This is really interesting. Higher numbers of CD8 T cells predict better outcomes and longer survival in a whole variety of cancers. And this has been studied extensively. This is an interesting study. I mean, these are in journals like the New England Journal of Medicine, you know. This is not like in the Khyber Pass Journal of Chiropody or something, you know. So these are serious journals. And ovarian cancer patients, this were 117 cases of standard ovarian cancer. Biopsies were taken at the initial surgery and they were frozen. That's important because you can preserve them for years and years and years and years. And then you can go back to them, say after 10 years, which is the general sort of format for these studies, and you can say, well, these patients are still alive, those patients have died, and several patients died after 1 year, 2 years, and so on, and try and correlate it with something in that sample which correlates with how long they've survived. And this is what's been found. In the high CD8 T cell group, and then there's the low CD8 T cells, the difference was the median survival there was 55 months, this is 26 months. That's double survival if you've got high CD8s. And sono and photodynamic therapy does that. It pushes up the CD8s in the tumour microenvironment, and that's massively important. Now, this is another very interesting study. Bilateral tumours. You've got an animal, you've got a skid mouse, for example, and you put a tumour on one side and one on the other. And then you do PDT or surgery on one of the tumours. You either do PDT or surgery on one of the tumours. The second tumour you don't touch. Now, in the untouched tumour, in the surgery group, there was no change in the immune response. In the PDT group, the PDT improves the tumour response almost by double. And it's action at a distance. What's the mechanism? That is cytotoxic CD8 T cells, which are tumour specific. That's what's important. Now, so PDT increases the CD8 white blood cells at the tumour sites. It reduces the number of metastases, secondaries, potential role for pre-surgical treatment of or vaccine production for biopsy samples, and it produces immunoediting. Benefits. Photosensitizers are non-toxic. So this really is something which is as close as possible to the kind of natural treatment approaches which is in all your hearts and what's been said today and so on. Targeted, no dose limitation. You can repeat it as often as you like. It is not immunosuppressive. Conventional treatment approaches are immunosuppressive. Vaccine-like response, action at a distance. Now, limitations, light penetrance. Red light only goes in a couple of centimeters. Sufficient light energy must reach the tumour cells in order to activate the sensitizer. And how do you get at that if it's deep in the body? Ultrasound, this is our solution. This is very, very clever, and this relates to the particular molecule we use. And because ultrasound will go straight through the body. And it's safe. Ultrasound was found to enhance the treatment effect of chemotherapy, actually, as in 1976. Later found that several photosensitizers also activated by ultrasound. One of the most interesting ones recently is an agent which has been developed by Provectus. The reason I know about this is that our group in New York, who are raising money for our mice series of patents, they raise funds for Provectus, and it's Bengal red. You know the stuff you put on your forehead, the red spot? It's Bengal red. Very, very simple, very non-toxic. Now, the mechanical effect on the sonar sensitizer, because where it sounds a pressure wave, produces free radicals, produces pores in the tumour cell, sonoporation, and cavitation, necrosis. Now, therefore, this is the agent we use, basically. We import it as an unlicensed agent under permission from the Medicines and Health Regulatory Authority. We have an import license. That's going back to what I first said. We work with the regulatory authorities because that's the situation we've got. So it's better doing that and working within that as much as one can. Otherwise, you can do nothing. That's the reality. Now, potential, it treats deep tumours, it's non-invasive, and it's targeted. Now, what is the evidence for ultrasound theory? Same sensitizer and ultrasound intensity as this case series. We did some animal studies. On the top line, we use the agent only. On the second line, we use ultrasound only. These are S180 sarcomas on mice, and we took the tumours off the mice and we measured them here. There's the top line, ultrasound here, then control. Now here, the ultrasound plus the agent, look at the difference between those. Those dots are small, those are big, basically. If you can't get your head around it, it's as simple as that, okay? So anybody can understand that. And the next, what about the amount of ultrasound you give? We found the best intensity is 1.2 watts. When you give less, you get less of an effect, and even less than that, you can get less of an effect again. Basically, that dot is smaller than that dot. Again, simple, okay? It's not even you can understand it, okay? It doesn't take a doctor, okay? So this is the histology. Let's not skip that one. Now, it also ultrasound will go into bone. We do a lot of work with tumours with bone secondaries, particularly breast cancer and prostate cancer, and we get effects on bony secondaries. We've done safety studies on zebrafish, standard model. This is basically, we tell our patients not to get exposed to bright sunlight for a few days afterwards, but we've never had that problem. Either that's because we get no sun in UK, which is probably true. There hasn't been any this summer, or even in very sunny weather, we've had no problems. With the porphyrin sensitizers, you get a lot of light sensitivity. And basically, our agent works in this 660 nanometers. The biggest effect on absorption is in the blue end of the spectrum up here, but blue light will only go in more, about 1/4 of a centimeter, so it doesn't have much penetrance. We use a light bed. This is a bed with probably about 50,000 LEDs. We also have some going at infrared frequencies at 940 nanometers. There's some evidence that this agent will respond to that as well, and that you can't see those because they're invisible. Now, this is the protocol. Sublingual administration, 48 hours on the light bed, then ultrasound, and with a 2-week cycle. And we add to that 1316 beta-glucans. A bit of practical advice for you, by the way. You need to go away from this meeting with something in your pocket, don't you? Now, use Imiflex. That's 1316 beta-glucans. I started into this many years ago with my experiences in China with cordyceps, coriolus, all that kind of thing. Those are 1,3,1,4 beta-glucans. Having the correct side chain is absolutely critical. This has been developed by a company called Biothera, based in Egon in North America. We've done some major papers for them. We've published one already, which is a trial of medical students. This is an RCT. We just completed another one this year on an older population, 60 plus. Green tree extract, pancreatic enzymes. I'm going to talk a lot more about pancreatic proenzymes in a minute. Omega-3s, really important, and some plant-based angiogenesis inhibitors and other relevant supplementation. The supplementation I use is based on looking at the patient from a TCM point of view. So those are organ-targeted preparations, but without going into the details of it, they do seem to work. Now, what about our outcomes? This is an example of some of our outcomes. The predicted median survival is red, the actual survival is green, the alive and exceeding predicted median environment is blue. And basically, in very simple terms, the green lines generally tend to be higher than the red ones, and the blue ones even higher. This is common across our cases. Look, these are end-stage cases. We're not curing everybody. So our treatment approach is palliative. Now, this is a case, non-Hodgkin's lymphoma, 60-year-old female, recurrent, it's a T-cell lymphoma, that's quite rare. Most lymphomas are B-cells. Resistant, partially response only to second-line chemotherapy. Also, high-dose intravenous vitamin C didn't work on her. She had progressive disease. Abdominal radiotherapy in 2005. She had 36 grays. That was she'd had a lifetime dose of that. Predicted median survival 6 months. Completed SBDT in July 2005. She's still alive and well and cancer-free today. That's absolutely astonishing. Okay, this is an example, like your mother, of similar cases. So actual survival, and when I wrote this, 41 months. It's now much longer. Brain tumor. 50-year-old patient presented in April 2008. Grade 3 ependymoma. It's quite a rare form of brain tumor, diagnosed in April 2003. First consultation, the clinical state was poor. Median survival 6 months predicted. Previous surgical debulking and whole brain radiotherapy. She'd had the lot, basically. All that was subsequently offered was temozolomide. Temozolomide, the standard chemo for brain tumors. It actually is reasonably effective, as a matter of fact. But anyway, she refused that. SBDT in April 2008. Covered by dexamethasone, because in brain tumours, if you get even slight swelling, you'll get fitting, basically. Outcome, a month after treatment, she felt well enough to go on a 2-month holiday abroad. She has remained relatively symptom-free. We did a further course in October 2008. She survived 10 months, alive and well. For a brain tumour, that's pretty damn good, I can tell you. So, and then repeated scans. These are scans without going into... This is your mother. 80-year-old female patient, inoperable non-small cell lung cancer, the left lung diagnosed June 2005, refused palliative radiotherapy. She actually had a bone secondary as well, presented in August 2005, given a predicted median survival of 6 months. SBDT completed in September 2005. Following treatment, she developed an 8 because that's where the bone secondary was. Stable disease detected in March 2007. June 2007, she was found to have tumour progression, so we did a second course. She tolerated this well, and she's alive and well today. How about that? So if you don't believe me, just have a word down here. Now, these are some of our non-small cell lung cancer patients. We've had very good results here. This is very interesting. It's because it largely down to the cancer immunotherapy aspects of SBDT. That's why we've had those good results. Another one, recurrent breast cancer, left side 1990, partial mastectomy radiotherapy standard approaches. June 2008, right-sided visual symptoms and severe eye pain. She had a secondary around the right optic nerve. Body scan showed some bony metastases and also malignant breast lump on the right side this time, too high risk for biopsy of the mass around the eye. She started anastrozole, then did SBDT. The pain in the eye reduced within 2 weeks and then fully resolved. Hospital visual fields assessment improved, breast tumour reduced in size, exercise tolerance and well-being increased, follow-up scan on orbit awaited, that was redone, that had improved. So that was, these are kind of really end-stage cases, you know, what the hell do you do kind of cases. Now we optimise this with innate immune function using 1,6-beta-glucans, tumour hypoxia, we use ozone. Ozone makes a big difference to our treatment outcomes because it raises the partial oxygen pressure in the tumour. We use ozone autohemotherapy. We take out 50 mL of blood and put in a measured volume of ozone and reinfuse. And then lastly, regulator T-cell modification. The regulated T-cells downregulate your tumour-killing CD8 T-cells. And so the best way of dealing with that is using very low-dose cyclophosphamide. Cyclophosphamide is a chemo agent. We use it at a fraction of the dose you would use it for chemotherapy. And that often helps a lot. A group of patients that I come across, which I found extremely challenging, are two sorts, sarcomas and embryonal tumours. They are all chemo-resistant. And for years I've been at this particular thing for about 7 or 8 years. And as my research degree was, my MD was in embryology, I was very familiar with the trophoblastic theory of cancer. Now, the trophoblastic theory of cancer is this. When the placenta embeds in the uterine wall, it has to be invasive, otherwise it wouldn't stick. Then, as the embryo matures and grows, it begins to secrete its own pancreatic enzymes. Now, what I realized, now this is a really important step, was that these enzymes are not secreted as ready-made enzymes. They're secreted as proenzymes, which are relatively small molecules, and we call them generically zymogens. Now, that was a major step forwards. And I looked at the research work done on this by John Beard. John Beard was professor of embryology in Edinburgh from about 1890 to about 1915. Most of his work was on fish and amphibians, and very solid scientific work. I was really impressed with it. As an embryologist, I could understand all of it. And I thought there must be something to this. So I thought this would work on these embryonal tumours and indeed sarcomas. Now, at that time, I had a female patient with an embryonal tumour in the bladder, totally chemo-resistant. The oncologists were raking her lots of chemotherapy, which is a complete waste of time. I told her to stop it. And I managed to get this made up, these pancreatic proenzymes, as a suppository by a pharmaceutical specials manufacturer. Now, there are about 30 specials manufacturers in the UK. The vast majority are completely and utterly useless, okay? I hope there's no pharmaceutical specials manufacturers sitting here. One of them was very, very good, and this was Mandeville Medicines. Mandeville Medicines is run by a very interesting chap, Karol Patzik. Karol's Polish, and he has all these, he keeps thinking the world's about to collapse and all the rest of it, which is kind of a bit like what Robert was saying and so on. But actually the world doesn't actually collapse and so on. And so I managed to convince him to do this for us. And I remember there was quite a lot of things stacked against me. I met him near to our rooms in London in Wimpole Street at the Institute of Directors in Pall Mall, and he was a member. And the night I got into London, it was snowing. Which is most unusual for central London. And I couldn't get a cab. So I walked to Pall Mall. It actually isn't that far. But anyway, I had this extraordinary meal with Karol, and he agreed to do it, and he did so. And this was at enormous cost. I can tell you we've been on the ropes a few times with this, but there we go. And so that was the start of it, and this led to this company called Propanc. You'll see some of our clinical results, and they were simply outstanding. And I thought we've got to do something about this. Now, this is actually a natural mechanism. So there's two problems. One, I had to fund it, and two, I had to patent it. That's the real world, okay? Whether you like it or not, that's the real world. So I figured out how I might be able to patent it, and I actually got it right. And I've got 5 patents on this. Secondly, I managed to raise some funding from shareholders. We formed a company, and we haven't spent a lot of money. We spent about 2.5 million so far on animal studies, cell culture studies, and so on and so forth, some of which we're going to publish. And we're now a public company, registered in America. Our main sort of market makers and lawyers and whatever are all in Manhattan. Do you know, they're amazing, the Americans. You know, with Hurricane Sandy, came over, floored everything. All our team were up and running completely in 3 days. How about that? What would have happened here in the UK? The Americans really know how to do things. I'm really impressed with them. And so that's where we're at. I actually raised all the initial funding on my own because I'm a practical kind of guy. And that's actually what works, okay? And we'll be able to take this through to proof of concept for relatively small amounts, probably another 10 million or so. And at that stage, in order to take it to phase 3, we have to sell out to Big Pharma because they're the only people who have the financial muscle to take this to the next stage. That's the real world. I don't get feeling the slightest bit guilty about that. I'm working with what's possible, and that's what's relevant here. Now, this is really important. This is why I think I found something that could be a real game changer. And this is from some tiny, insignificant clinic in the middle of the countryside in Hampshire, that's all. Now, in embryogenesis, I'll read it from here. During gastrulation, the cells peel away from the ectoderm and migrate towards the center of the embryo to form the mesoderm. That's embryology. Now, this is the important thing. This is one of the most important questions in cancers and in sarcomas, and that's the ectodermal to mesodermal conversion involves a process called Epithelial-Mesenchymal Transition. This is a change in phenotype, not in genotype. Phenotype is the way cells behave. Genotype is the software in the cell which exists physically. But the way the cells express is called a phenotype. You can do something about the phenotype. You can't do anything about the genotype. Now, once cancer cells undergo epithelial to mesenchymal transition, they become motile and they move away and they become invasive. Now, the most important molecule here is a molecule called E-cadherin. It's a sticky molecule, and that's absent in cancer cells, and it's essential to the epithelial-mesenchymal transition, and this is central to the process of cancer stem cells and also sarcomas. These tumors, all tumors, are driven by stem cells which have gone nuts, basically, and they've undergone an epithelial-mesenchymal transition. Chemotherapy does not kill those cells. This is why chemotherapies rarely produce a cure. Cancer immunotherapy can do something about that. It looks like this can do that as well. Now, this is the science of it. Basically, this is the sticky molecule, E-cadherin, and this combines with beta-catenin. Without getting into the detail of that, because it'll blow your brains out just before lunch, just let's come on to what we did. Now, we did some studies on some standard tumor cell lines, OE33, which is an esophageal cancer cell line, PANC1, which is a pancreatic cancer cell line, and we upregulated E-cadherin massively, and we killed all the tumor cells in the cell culture. What we also found, and this is the most interesting thing, is that in our pancreatic cancer cell lines, these tumor cells started to secrete insulin and glucagon. Look, when you get pancreatic cancer, you don't become diabetic. That's because the whole of the pancreas isn't taken over by the tumour. But the tumour cells themselves are very abnormal in the pancreas, so they no longer secrete glucagon or insulin. We've managed to show that they will do that. This supports the idea that we're working on this really important transition, the epithelial-mesenchymal transition. And as we speak, we've just done some further studies on that. This for a long series of reasons. We're doing this at the University of Granada in Spain, where the Alhambra is, and so on. And we've just sent off our gene chips on our last cell culture studies to look at the EMT genes. So, but I think we're going to come out positive on this. Right, this was the case that I first worked on. This was the embryonal tumour. She had a urachal tumour. Pancreatic proenzyme treatment commenced in mid-September 2007. Treatment continued until her death in August 2008. She survived much longer than she was predicted. Another case here. This was a colorectal cancer. The life expectancy was 6 months. She survived for 17 months. These are the various studies which I found from the turn of the century using fresh pancreatic enzymes. Now, they had to be very fresh, and these were injected, but it was the proenzyme bit that had the effect. It was quite clear that this is because we understand a lot more about enzymes now than we did in 1900. Now, one of these cases was a fibrosarcoma. What I do know with this treatment approach with proenzymes we've been using, one was an angiosarcoma, one of the most aggressive of sarcomas, and that particular patient is tumour-free after nearly 5 years, and that's truly amazing. Sarcomas express an extreme of the epithelial-mesenchymal transition, because sarcomas are very what we call mesenchymal. That's the science of it. And then we've done some angiochamber studies where we look at angiogenesis inhibition. We found that these tumours, secretion of vascular endothelial growth factor, which causes tumours to grow, it downregulates that. So basically, we're getting a whole pile of information from the laboratory and from animal studies. We've done a huge number of animal studies on skid mice. That's seriously compromised immunodeficient mice. We've done those in Australia. At one stage, I bought every skid mouse in Australia. How about that? That's quite an achievement. And so hopefully now, as the people in New York get going from us, we'll raise the next amount of money, and then we'll start phase 1, phase 2 clinical trials, because we've found specific bits of each proenzyme are really important to have maximal effect. And those of you who can read between the lines, we have permission to carry out phase 1, 2 from the Medicines and Health Regulatory Authority in UK. UK is the last place I'm going to do this. I'm going to do these studies in Germany and Switzerland. I could explain that privately to you in due course, but UK is the wrong place to do it. And so that looks very exciting. Now, the whole point of this is that this last thing is maybe a major breakthrough. It just comes completely left field from a slightly nutty doctor like me, and it might have a big effect because it'll end up being cheap, okay? And it would be nothing like the monoclonal antibodies, and it will be effective over a whole range of tumours. And then that comes back in to where I started, where you all started, the Cancer Act 1939 and so on and so forth. Now, there are two sides to this, and you've got to listen carefully to this because this applies to you, okay? All of you who have got anything to do with cancer. Right, two sides of the equation. There are complementary therapists who are doing this, that, and the other, nutritional therapists and so on and so forth, doctors like Stephen and whatever, and we maybe use acupuncture, whatever it happens to be. And we treat patients with cancer or whatever wording you've come up with. And then there's the other side of the equation, who are the oncologists, who use very standard things. The treatment protocols are usually surgery, chemotherapy, and radiotherapy. These often work, and they absolutely do. And so, what are the ups and downsides of both of those? Now, I see a lot of patients who have seen various complementary therapists, and basically they have essentially given the message that don't do any chemotherapy, radiotherapy, or surgery. They're dangerous conventional treatments. And as a result, these patients have been given bad advice, in my view, and many have died as a result because they have not been properly informed. Wasn't that complementary therapist did not have an evidence base to say that using arnica at some centesimal, some LM dilution or whatever is going to cure them. They've, at one level that's bollocks, but it's, it just doesn't stack up. Now the important thing in a case that's coming to you who doesn't have a life-threatening illness, this is not so important, but if somebody does have a life-threatening illness, you must receive advice at some stage with a doctor who knows conventional oncology, which I do, and conventional radiotherapy. You should know all of that, and also have some knowledge of complementary medicine. Those two kinds of disciplines ought to be brought together. And we're trying to do that through the British Society of Integrated Medicine, because that will mean that patients will be better informed and may be able to make those choices, which could have huge, important repercussions in their own lives. Then there's the other side. There's the oncologists, bless them, who many of them are vitriolically opposed to all of these approaches out of usually total ignorance, and they can't quite see it from that point of view. So in other words, I'm suggesting there's an argument for both sides, and trying to bring them together in the middle, integrating them, leads to a better outcome. German oncologists are much more open to this approach than the UK oncologists. I've had a lot of interest from Germany and indeed Switzerland with what I'm doing. No interest from UK oncologists. That's why I'm doing my clinical studies in Germany. If you've got any brains, that's what you would do. And I think I've got a few of those. Now, by the way, the implication in all of this, with the oncologists at one side and all your complementary therapists and nutcases like Stephen and so on and so forth, is that the oncologists are really intelligent because they're doctors, and we're really rather stupid. It reminds me of something that happened to me during my surgical training. I was a demonstrator in the part of anatomy, and we used to examine for second MB. And this student came to me, and second MB anatomy, and I gave him a femur, and I said, right, Jones, tell me what this is. And he said, sir, this is a bone. And I said, I know it's a bone, I want to know what bone it is. And then he said to me, look, sir, I only want a pass, I don't want a distinction. I passed him. And then another thing springs to mind. There was something that I happened to see on Channel 4, they have this 5 minutes thought or whatever it is after Channel 4 News. That Stephen Isserlis was on. Stephen Isserlis is a very well-known professional cellist. His wife was a violinist, and it rang a bell with me because I used to be a professional musician, and I played with his wife in several orchestras and whatever, and I didn't realize she'd died. She actually died of metastatic breast cancer, and what Stephen Isserlis said, and this is from the patient's point of view, he said he wished that the various chiropractors and naturopaths and whatever, which his wife liked to go to, had said to her that don't do chemotherapy, don't do radiotherapy, and he thought that down-regulated her survival chances. Do you know he was right? So he has a point to make. So that's it. Thanks for listening.